Good morning, everyone, and welcome from London. I'm delighted to be here this morning, and I'm delighted to introduce from London Dr. John Rohrer and his FTD team. So with John today, we should have Dr. Lucy Russell, Dr. Martina Bochetta, Dr. Itana Sogov-Estev, Misha Clark, and Emily Todd. So thank you for joining us today on an early Saturday morning. Dr. Jonathan Rohrer is the consultant neurologist at the National Hospital of Neurology and Neurosurgery here in London. It's a leading hospital for neurology in the UK. John is the FTD lead at Dementia and the clinical rare dementia support, frontotemporal support groups. It's an absolute delight to work with John and his team at the Dementia Research Centre. And this morning they'll be spending an hour on talks from the whole team, including international collaboration across the world and biomarkers and FTD. They're also going to be doing a Q&A session afterwards. So please get your questions in, and John and his fantastic team will aim to answer them. Over to you then, John. Thank you.
Well, thank you very much. We've got an hour or so, and we're going to talk a little bit about FTD research in the UK, but also the importance of international collaboration. We work at the Dementia Research Centre in Queen Square. I'd just like to draw your attention to our website, which is www.ftdtalk.org. There are lots of really helpful fact sheets that have been developed in tandem with both caregivers and people that have FTD. So I hope people find that helpful. There's also some updates about our own research there. But I'm not going to do much talking, at least until the Q&A session. So I'm going to hand over first to Martina, who's going to tell us about the large project that we lead from the UK, which is the GEMFI project.
Thank you, John, and good morning, everyone. So today I will be talking to you about the Genetic Frontotemporal Initiative, also known as GEMFI. So now, FTD is a very complex and heterogeneous condition where different people can experience different symptoms. So someone might experience changes in their behavior, while others may experience difficulty in their language or associated motor disorders, while others can also have a mix and a combination of all these symptoms. There are also different types of abnormal protein that can accumulate in the brain. So this is a very variable condition. Now, the big question is, next slide, please, John. The big question is, what is the cause of FTD? And the answer is that the only known risk is genetic. And this will be the case for about 30 to 50% of people living with frontotemporal dementia. The cause for this is a mutation, so a defect in a gene. And the most frequent genes that are involved in genetic frontotemporal dementia are called CNANOR72, MAPD, or progranulin. But there are other rarer genes. Now, what does this mean? It means that if a child is born with a mutation, it means that unfortunately they have inherited this mutation from one of the parents. And at some point in their life, they will develop the symptoms of FTD. However, there is a window of time before the onset of symptoms where it is possible to detect and measure changes that occur in the brain. And this is extremely important. We can understand, by investigating these biological changes that occur in this window of time, we can better understand what is the progression of genetic FTD. And also we have a window of opportunity where we can intervene, where we can treat people before they experience symptoms. But we need to accurately measure how these changes occur. However, there are some challenges with genetic FTD. The first is that genetic FTD is a rare condition, and its estimated prevalence is about 3 over 100,000 individuals. And this is considering all age category. Now here you can see that these genetic mutations are much more frequent in C9orf72 overall. And you have here an idea of the number of people living with genetic FTD across different countries in Europe and North America. Now, the second challenge is that not only FTD is rare, but there is also a large variability. Next slide, please. There is also a large variability of where people with this mutation live across the globe. So, for instance, if you look at Spain and Italy, the most frequent cause of genetic FTD is granulin. If you look instead of the Netherlands or the west coast of America, the most frequent type of mutation is in the MAPT genes. Or if you look at the UK or Australia, it's rather C9orf72. So how can we solve these challenges? So the answer is we need to work together. We need to join forces and bring together centers with expertise all around the globe to study genetic FTD in a very uniform way. And we need to get ready and prepare for trials. Now, there are several initiatives all around the world which have started independently, but have now joined forces together. And Lucy will tell you about them later on. But let's focus on GENFI now, which was the first initiative that was launched. Next slide, please. So GENFI is coordinated by University College London, and it started in 2012. It started with 5 centers in Europe and Canada, but it grew very quickly over the years. Now we have 27 centers who collaborate together, and we are aiming to have more than 30 in the next phases of the project. It is a large multi-center consortium with the aim of tracking the evolution of genetic FTD, with the idea of using a common platform and a standard protocol among centers. What it means is that centers have agreed to work together to collect data in a very similar way, in a systematic way, to share and join expertise in order to research different aspects of frontotemporal dementia. Next slide, please. GENFI includes participants from different stages of genetic FTD, and that's to study and be able to understand what is going on at the different stages of the condition. So we include individuals who have symptoms, so a diagnosis of genetic FTD, and with a known mutation linked to frontotemporal dementia. So these are either mutation in the genes MAPT, C9orf72, granulin, and recently we started including some of the more, the other rarer form of mutation. Now, participants can also be their first-degree relative who are defined to be at risk, as they have 50% chance of having inherited the mutation. And if unfortunately they will be in a stage where they have not yet developed the symptoms, but they will. Next slide, please. So far, there are over 1,100 participants who have very generously dedicated their time to come for research visits. And that's since 2012 when the project started. This visit happen every year, and that's really helpful because it allows us to see how things changes over time. And these visits consist in a clinical assessment together with cognitive and a neuropsychological test. And we also acquire a magnetic resonance scan, which is truly important to measure what is going on inside the brain. And also we collect samples, biological samples, such as blood or cerebrospinal fluid. So all these data are and will be analyzed with the aim of developing indicators of changes, ideally with the earliest changes, because we want to understand what is happening very early on in the FTD. And we also want to track progression of frontotemporal dementia, which is so very important for clinical trial and for their design. So at the end of the day, what we want to achieve is to get the best scientific evidence to give the best opportunity to our patients and family to finally find a cure for frontotemporal dementia. So with this, I just want to thank all the participants, carer, and family members who take part in GEMFI all around the world. And in the next slide, you will see all the names of the investigators in the GEMFI centers. And if you want to know more on getting contact with us, please do have a look at the GEMFI website, which is www.gemfi.org. And thank you for listening.
Thanks, Martina. The slide with everyone involved is now right at the end, but I'll show it when we get towards the end. So, Lucy, you're going to move on and tell us a little bit about what Martina has already prefaced, which is this combination of GEMFI with the other studies around the world.
Yeah, thank you both. So, good morning, everybody. I'm going to tell you a little bit about the Frontotemporal Dementia Prevention Initiative. So it's also referred to as the FPI. And this is made up of a collaborative group of people, including many of the large FTD research groups across the world. So this includes GEMFI that Martina was referring to earlier, as well as a group in America known as World FTD, as well as some groups that have recently joined us in Australia called DINAD, and in New Zealand called FTD Gens, as well as a group in Latin America called RedLat. So we're continually expanding. We're all aiming to collect data in a similar way. And we're also reaching out to groups in Asia as well. I've been in touch with a few people from India and Japan and Indonesia about coming on board with the FPI. So we're also joined by many consortium groups from across the world, such as the World FTD United, who have kindly organised this event here today. So the hope is that we'll be able to bring together FTD research from across the world to promote clinical trials of new FTD therapies. Our initial efforts will be focused on individuals with families who are known to carry the genetic mutations that Martina was talking about earlier in C9orf72, progranulin, and tau. So other familial and sporadic forms of FTD are also of interest, but our first steps will be in these three genes, as we know more about the cause of the illness than we do in these other cases. So the main aim of the FPI is to connect familial FTD studies from across the world in order to move forward clinical trials in the field. So a key goal is to share information on genetic FTD clinical trials and work together to ensure that participants have the best opportunity for trials. We're wanting to promote responsible data sharing and clinical trial standards. So in order to do this, we are in the process of developing an international database of familial FTD research participants. And within this, we'll be able to identify those who may be eligible for upcoming trials, as well as using this database for larger group projects to improve the reliability of our findings in our research work. So moving on to clinical trials, I'm going to touch a little bit on the stages of clinical trials before we launch into the trials that are currently underway in FTD. So there are 5 phases of clinical trials. Now, the first phase is a preclinical stage in which pharmaceutical companies are developing their drug, and they must demonstrate that there is a good reason to believe that the drug may benefit people. This isn't done in humans, it's rather done in labs. And once the drug has been established, which can take many years to do, it then moves into a phase 1 clinical trial. So the phase 1 trial is made up of 2 parts. The first part is called a first-in-human trial, and this is usually given to healthy individuals to make sure that the drug isn't detrimental to humans at all. And then the second phase is a phase 1b, which is the first time it is given to the target population. So in our case, either individuals at risk of developing FTD or those who are symptomatic and currently suffering from FTD. So the aim of this phase is to determine what the maximum dose of the drug would be, so how much to give, and to monitor the safety and how well tolerated the drug is, and note any side effects that may occur. These trials are usually very short, from a number of days up to 8 weeks long, and only contain a very few number of participants. So if this all goes well, we move into the phase 2, which is a larger trial ranging from maybe 100 to 300 participants at most. And the aim of this phase is to work out if the drug works or not, while still monitoring the safety and the side effects of the drug. So this phase can take around 1 to 6 months to complete, and the entrance criteria up to this stage is often very strict. But when we move into the phase 3 trial, this is where this is classed as the pivotal study to determine if the drug does what it says it should do. And they're often on much larger scales, up to 1,000 participants. And in order to establish if the drug is successful, there's many different outcome measures that we must consider. And my colleague is going to talk to you a little bit about some of these outcome measures later on. So if we get to this stage and the drug is successful, we move into a phase 4 in which it's rolled out to all individuals who may be at risk or the target population, and then they're monitored over time. So takes into account a lot of individuals who are taking the drug in an observational study just to see how people are getting on. So the clinical trials in FTD, there's a number of trials in the pipeline, and we're in contact with over 10 different companies that are trying to develop therapeutic drugs, but all are at different stages. So at present, most of the FTD drugs are in the preclinical stage. However, there are a number very close to starting their phase 1 trials, as well as one currently in the phase 2 and about to begin their phase 3. So for both tau mutations and C9orf72, a company called Biogen are aiming to develop what is known as an ASO. So this is an antisense oligonucleotide. So it's basically a gene silencing drug. So it's aiming to turn the faulty gene off. And in these 2 mutations, because the gene is producing too much protein, which we think leads to the illness, we're hoping that the drug will just prevent this overproduction and bring the levels back down to normal. The drug will be injected via a lumbar puncture into the lower back, and it makes its way up to the brain, hopefully finds the faulty gene and turns it off. Biogen are currently using this type of drug in a group of individuals with motor neuron disease that carry the C9orf72 mutation, but are awaiting the results of this study until we know if they will be taking it forward in frontotemporal dementia. Another company called Wave are also working on this, and hopefully will be up and running next year. So the moving on to progranulin, there have been 2 different methods that have been employed to try and treat frontotemporal dementia with progranulin mutations. And the first one is known as a monoclonal antibody. So in progranulin, we believe that there's too little of the protein being produced, which causes the illness. And so this type of drug is aimed at stopping the body from breaking down progranulin, as you can see here in the image on the right. And the drug is administered by injecting it into the bloodstream, just like you would if you were giving a blood sample. And a company called Electa is sponsoring this trial, and their phase 1 study was successful. And so they moved on to phase 2. And interestingly, the results from their phase 1 trial suggested that not only could they increase levels of progranulin, but by increasing the dose of the drug, as in how much is given, the levels of progranulin at the end of the trial also increased. So very exciting results. And we're looking forward to seeing what the results of the phase 2 study are. The second method used in progranulin is what is known as inactivated virus technology, and it's called adeno-associated viruses, AAV. And this takes a healthy gene and puts it into a virus, which is injected into the body using a lumbar puncture needle. But instead of in the lower spine like before, this time it's going into the back of the neck. So it's a little bit more invasive, but the procedures only have been given once, whereas in the Electra trial, you have to receive the drug multiple times. The hope is that the virus will be release the healthy gene into the brain and hopefully increase the levels of progranulin being produced. There are currently 2 companies working on this, Prevail and Passage Bio, and both are hoping to get started next year. So just to summarise, really, there's lots of work going on behind the scenes to develop therapeutic drugs for frontotemporal dementia, and hopefully the Electra phase 3 will be up and running very soon, with a couple of other trials starting sometime next year. So thank you ever so much for listening. If you do have any questions, please let us know. You can also find further information at www.thefbi.org.